In my day job, I am a doctor who treats severe respiratory failure, and one of the most common enemies I encounter is pneumococcal pneumonia, Pneumococcus, a very common bug, and probably the most common cause of pneumonia, certainly in the United Kingdom. This slide is showing 3 air sacs, 3 alveolar subunits in the lung. So we're right down to the end of the lung where gas exchange is occurring. The alveolar air sac in the middle, stained yellow, is infected. It's an isolated unit infected with pneumococcus, and you can see individual bacteria in the centre of this alveolus. The 2 sets of alveoli to the side are healthy and normal, and you can see normal gas exchange there. The final bit you can see in the infected alveolus is these purple neutrophils, which we've discussed this process before in the first series of slides. They're invading, they are recruited to that area as they're trying to deal with the host infection. Now, pneumococcus is very clever. The first thing it does is it actually stimulates proteins on the pneumococcal surface, can actually directly stimulate net release from neutrophils. It also produces a protein called pneumolysin, which is directly toxic to neutrophils and can leads to their death and destruction too. So it's manipulating neutrophils in 2 ways. As you might imagine, if you're stimulating neutrophils, you get net release. But pneumococcus can degrade nets. It uses an enzyme called endonuclease to do that. And here you can see these green enzymes breaking down and chopping up nets. The bacteria can consume those components as fuel, as feed to keep it going, but it also enables it to escape that net, that trap. It enables it to escape the immune response. So it's directly manipulating, again, it's an example of a bacteria manipulating our immune response to its own advantage and is gaining a survival advantage through that. On the other side of the graph, pneumolysin is leading to neutrophil apoptosis. That's the death of the neutrophil. When these neutrophils die, they release the enzyme neutrophil elastase, which is an important component of nets. And this neutrophil elastase can break down that nice intact endothelial barrier, which leads to degradation of surfactant, this key protein which helps keep our lungs open, and destruction of the extracellular matrix, and destruction of key proteins in that membrane too. And so ultimately, our air sac, the wall of our alveolus, gets degraded and breaks down. And so you can see how this impairs gas exchange because you've just damaged or destroyed the integrity of that air sac. You can see how it facilitates pneumococcus escaping because it's damaged that air sac and it can escape, and it leads to quite a profound inflammatory response. And that is kind of what's happening with pneumococcal pneumonia. The final thing that it can do is neutrophil elastase actually can impair another key cell of the immune system, macrophages, and so that impairs the action of macrophages, sort of another cell in the orchestra of our immune system trying to fight the infection. So what we're doing, we're saying that nets are really important in the immune response, but also they can be manipulated, they can be hijacked or distorted, their function can be distorted by bacteria and viruses to support their own nefarious purposes. I've put this series of slides in just to show how important nets are to the immune response, and actually how you know they must be important as microorganisms have developed a series of strategies to overcome them, subvert them, and get around them. This short table just shows a list of a number of ways that various microorganisms have used to escape and get round nets. Classically, they chop them up and break them up so that they can escape from them. But some, such as plague, Yersinia pestis, can use different proteins to suppress the production of reactive oxygen species. If you go back to the first talk in these series of lectures, we talked about neutrophils having 3 functions, and the very first purple circle picked up on the reaction of or release of reactive oxygen species from neutrophils as part of their key killing mechanisms. And you can see how bugs are, Yersinia is particularly manipulating that in this example. TB can also affect the release of enzymes and then hydrogen peroxide. Hydrogen peroxide is really important in neutrophil trafficking and neutrophil signaling. So I won't go into the details of this table too much, but many bugs have evolved to manipulate and distort our immune function to their own advantage, and distorting neutrophil extracellular traps is a key part of the survival advantage they're leveraging over their competitors. So in this short series of slides, we've covered how bacteria and viruses can manipulate neutrophil extracellular traps. And really it's just trying to build these blocks together to explain the interaction between microorganisms and the key components of our immune system. Immunothrombosis is a host defense mechanism where inflammation activates the coagulation system to limit the spread of pathogens in the bloodstream. And that's what you can see occurring in this slide. You have activation of neutrophils, complement activation of platelets, complement activation of neutrophils, neutrophils talking to platelets, platelets talking to neutrophils. You can see at the very top how the pneumococcus has stimulated monocytes and degraded their action, how it has damaged the lining of the air sac and leaked into the microvasculature and the whole host of the immune response here. When this pathway, or when immunothrombosis becomes uncontrolled and excessive, when you get a dysregulated response, immunothrombosis progresses to thromboinflammation. And this excessive thromboinflammation causes tissue damage through microvascular thrombosis. So you see patients with sepsis progress to multiple organ failure. This was particularly severe in COVID-19, but is seen in other infections like pneumococcus that we've used as an example here. The model of immunothrombosis is extremely powerful. It bridges the immune response from a cut, an abrasion, a penetrating injury, and how we respond to an invasive pathogen, a viral chest infection or a respiratory infection. It's activation of our innate immune system and its coordination together. This is an incredibly localized, incredibly controlled process, or immunothrombosis. Right at the heart of this is H3.1, as we now know that nets act as a critical scaffold to support the immune response to infection for both bacteria, viruses, and fungi. This is actually it in the real world. This is a picture of the lung of a mouse, and this mouse has been infected with an organism called methicillin-resistant Staphylococcus aureus, MRSA. If you read the Daily Mail, MRSA is one of the superbugs. The first panel, panel A, is just picking up neutrophils being recruited to the lung to the site of injury. Panel B and panel C are actually picking up nets there. So it's not just a model, this is actually what's happening in situ. So C, this sort of light blue colour, or greeny blue colour, is cytox green, and that is picking up DNA that's been released from neutrophils as they've really drilled down in high resolution into what's happening there. And so you can see MRSA is stimulating neutrophil recruitment and stimulating net release in the alveolar lining of the lung. And that's actually what we actually see down a microscope in real time in the lungs of mice. And that's sort of trying to bring these models that we've put together to show you this is it actually in tissues. Again, you've seen this slide before. This is the pink mist behind stimulated neutrophils which are erupting. And again, the point here is that the scale is at the bottom. Those capillaries are sort of 20 to 40 microns across, and you can see 50 microns long, some of these tails, perhaps even more, and how the nets lead to the complete occlusion of these microcapillaries where oxygen exchange and nutrient exchange is occurring. Again, so it's just trying to build this rich picture of the immune response and describe it in real time. This beautiful electron micrograph is actually showing at the top and at the bottom there's the airspace. And what you can see is a red blood cell squeezing through and being bent in half to flex through the tiny space of this capillary. And what you can imagine here, that these white cells, neutrophils, when they're activated, are just blocking these microcapillaries. And that's really what we're just trying to show. In the top left, you can see the incredibly thin basement membrane and endothelial cell. And that's the pictures we've been trying to put up, and forms a key part of the model that we've been explaining.